show of hands, who has done the policy acknowledgement? 47 out of 47. That is what I like to see. That is consistent with what I see in the grade book. You can translate that. That is consistent with, I see the grade book most people have done it already. That's great. If you haven't done it, just like do it today after class. Just get it done. Make sure that it's not a problem. The due date is Friday, 20th at noon, so you have to have that done. Second thing is that we didn't finish last time, which is a theme in this first couple of weeks of the semester historically, so we are keeping that tradition alive, which is nice. So this is what we're going to do today, but we're not there yet, so we're going to finish part 4 from last time, the emergent properties of water. So we're just going to pick up your notes right there, and after we do that, we'll come back and we'll do biological molecules. One more quick note before we get into things is just remember to go to workshop this week, because workshop starts this week. So on your schedule, you have 2 periods of biology that are in this room, Tuesday and Thursday afternoon at 3:50, and then you have something else at a different time, a different place. That's your workshop. Make sure you go this week. The only thing you need to bring this week is a blank bios form and your notes on paper. I promise you the bios form will make more sense after your first workshop. So let's pick up here. This is the last thing we saw last time. Now I know it's late in the afternoon and everyone starts to get a little sleepy around this time, but also it's a lot easier to see the screen if I turn the lights down just a little bit. What would you prefer? Keep the lights up or turn them down a little bit? What do you?
Down.
Yeah, let's do that. That's doing nothing. We'll turn that off and then there we go. That's a little better, it makes it easier to see. Okay, so this slide is reviewed. We did this last time, but just to very briefly review the concept of a hydrogen bond. It's a type of van der Waals interaction, so it is between 2 different molecules, and it occurs when you have a partial positive charge on usually a hydrogen, and that partial positive is going to associate temporarily with the partial negative charge on usually it's either an oxygen or a nitrogen, some highly electronegative atom. So that interaction right here is a hydrogen bond. I want to be very clear because this is a common kind of place for confusion. This bond right here, the oxygens and the hydrogen within the water molecule, that's not a hydrogen bond. That is a polar covalent bond. The hydrogen bond is what occurs between 2 different molecules. Right, so that's where we finished with last time. So today we just need to finish up this part 4, then we'll get into the new stuff. So looking at the emergent properties of water, the first emergent property of water is hydrogen bonds. Basically, water is polar, as we've seen, due to the OH, polar covalent bonds, and that means that the molecule has regions that are partially positive, regions that are partially negative, and those regions will be attracted to each other. Okay? So within liquid water, you have just a constant forming and breaking of hydrogen bonds. Each water molecule can form 4 bonds. At any given time in liquid water, most of the time, they're not making all 4 at once, because the water molecules are in constant motion. Some of them are going to be too far away, some of them are going to be too close. So you can make up to 4, but oftentimes it's 1 or 2 at any given time, each water molecule in a container of liquid water. Now this matters because all the other properties we're going to see are due to the hydrogen bonds that water can participate in. So we're going to go through 4 more properties here. Again, it's all due to this. So the first is cohesive behavior. There are 2 types of this. First is cohesion, which just means the water molecules stick to each other. For example, in like the transport tissues of plants. So water is moving from the roots up through like the trunk of a tree. Water molecules stick together. That's called cohesion. This also causes surface tension, which is where you have the surface of water. The water molecules on the surface tend to all be making H bonds with each other. So you make kind of a film on the surface of the water. You can see that illustrated right here as this swimmer kind of breaks through. You can see the layer of water that's covering your whole head there. And this film actually can support a little bit of weight as long as it's widely distributed. So you can see like a spider, or you often see insects that kind of walk on the surface of water. That's possible because of the surface tension. And the surface tension exists because of the cohesion between water molecules. So it's a fancy way to say water molecules are attracted to each other. It's called cohesion. So what do you think the next slide is going to be? It's going to be adhesion. Adhesion is the same thing, except it's water molecules attracted to other things. And we can look at plants again for an example of this, because it's not just that the water molecules as they're being transported are sticking to each other, they're also sticking to the walls of that transport tissue in plants. So that as the water moves up, it sticks a little bit, it doesn't fall back down at any point. Another example of adhesion that you may have be familiar with is capillary action. So this is like if you've ever had like some water spilled on like a table and you put a napkin in it, but like the napkin or the paper towel only partially overlaps the water, or the water kind of like gets sucked into the paper towel. You can even do this holding the paper towel vertical, and it will work against gravity. That's capillary action. So as long as the material is what we call hydrophilic, just means it'll associate with water molecules. We're going to define that more specifically in a minute. As long as the material is hydrophilic, it'll experience capillary action. And again, that is due to adhesion, the attraction between water molecules and other things that are capable of forming hydrogen bonds. Not everything is capable of doing that, but when it can, you can have adhesion. Okay, so that's cohesive behavior, cohesion, adhesion. Yeah. I can try to slow down a little bit. I'll try. Are we okay with cohesion and adhesion? Are we all right with that? All right. Our next emergent property of water is that it moderates your temperature. The reason for this is that water has a very high specific heat. Specific heat is a very particular thing, and we're going to talk about this more next semester when we talk about nutrition, we'll talk about like calories and all that stuff. So we'll get into this more next semester, but the specific heat of a substance is how much heat is needed to increase 1 gram of that substance by 1 degree Celsius. That's the specific heat of a material. Increase 1 gram by 1 degree Celsius. Anybody know the specific heat of water? It's 1 joule is a good guess, not quite 1 joule. That's a really good guess. It's 1 calorie. You don't need to know this, by the way. You don't need to know that it's 1 calorie. Like, that's just fun fact. But think about what increasing the temperature of something does. Right? When you increase the temperature of a material, what's actually happening at the molecular level is that the atoms and the molecules are moving faster. Right? You put energy into it, stuff moves faster, and that heats it up, right? That faster movement is heat. Now for water, we have H-bonds, right? We have cohesion. That means the water resists moving faster. You can put energy into it, but if that energy isn't sufficient to overcome the H-bonds, the attraction between the water molecules, then that energy won't actually cause the water molecules to move faster. So it takes a lot of energy to actually change the temperature of water. You have to put a lot of energy in to actually increase that temperature by 1 degree Celsius. And again, the specific heat for water is 1 calorie. This is a fun fact. You don't need to know this. We're going to talk about like what that means next semester. It's 1 calorie per gram per degree Celsius for water. Takeaway is it takes a lot of energy to increase the temperature of water. So that's like, okay, that's cool and interesting, but why does this matter? Turns out this matters for a lot of reasons. We're going to look at 2 of them. We're going to look at a big scale reason and we're going to look at a small scale reason. The large scale reason is climate. And Southern California is the perfect illustration of this. along the west coast of the United States, the ocean current is a cold current that's coming down from the Arctic. So it's a big circle in the Pacific Ocean, right? And I guess for your orientation, it's this way. So you're coming down from the Arctic, from like Alaska, you're coming down the west coast of the United States. That means that this water is a lot cooler than the atmosphere in this area. So the cities that are close to the coast, like LA or Santa Barbara, San Diego, nice and cool, 73, 75, 72, beautiful. But if you go inland just a little bit, you lose that cooling effect of the ocean. The ocean is absorbing that atmospheric heat. That's not happening in here. So Burbank, 90, 96 for Riverside, 106 for Palm Springs. The atmosphere is not fundamentally different between these places. They're just a few miles apart. The difference is the proximity to the ocean that can absorb all that heat from the atmosphere. This is really important. This is because of that high specific heat that water has. We also use this to cool our bodies. It's called evaporative cooling, which is just a fancy way of saying you sweat. When you sweat, your sweat glands release a little bit of water onto your skin. The water droplets sit on your skin. Now your skin, most of the time, is going to be warmer than the atmosphere. So the water on your skin absorbs heat from your body. If it absorbs enough heat, it evaporates off your body and it takes the heat with it. That only works because water can absorb a lot of heat. It only absorbs a little bit of heat, and it wouldn't remove heat efficiently enough to be a useful cooling mechanism. But because water has a high specific heat, we can actually use it to cool our bodies. That's what we do. That's what sweating is. Okay, so that's our next property, was the high specific heat and the implications of that. Next, we have expansion upon freezing. This is a really interesting one. Normally this doesn't happen. Normally the solid is more dense than the liquid. For water, it's the opposite. The liquid is more dense than the solid. And the reason for this is the H bonds. So in liquid water, only about 15% of the water molecules at any given time have 4 hydrogen bonds associated with them simultaneously. You don't need to know that number. I'm never going to ask you that. The reason it's only 15% is that in liquid water, many of the molecules are actually too close to each other to form H bonds. The distance for an H bond has to be just right. Too close or too far, it won't work. Now, as water freezes, you lock in those hydrogen bonds. So by the time you get to solid ice, every water molecule that's part of that ice crystal is locked into 4 H bonds. As a result of that, the average distance between the molecules in ice is greater than the average distance between molecules in liquid water. This means ice is less dense than liquid water, which means it floats. And this means that a body of water freezes from the top rather than the bottom. So think about like a frozen lake in the winter. I have a friend who lives out in Canada, in Saskatchewan. He goes ice fishing in the winter. So they go out in the middle, I don't know why they do this, it sounds terrible to me, but he loves it. They go out in the middle of the lake and they drill a hole through like several feet of ice, and they fish through the hole. There's liquid water down there, there's still fish, and they're fine. Why does that happen? Because as the ice crystals start to form, as the water molecules lock into that crystal structure, they become less dense and they float to the top, so it freezes from the top. You can preserve the ecosystem down below. If this wasn't the case, if it was the opposite way, bodies of water would freeze from the bottom up, and every time like a lake froze, it would be a solid block of ice. That would kill everything every winter, and you couldn't have lake ecosystems in places where they froze. So this is a really important property for just like how ecosystems function. This is a really big deal. The fact that ice floats because it's less dense than water. All right, we have one more. And it is versatility as a solvent. Water is sometimes called a universal solvent, which is not entirely accurate. Strictly, not universal. But a lot of things will dissolve in water. Anything hydrophilic, which just means it has an affinity for water. What things have an affinity for water? Anything with regions of partial positive and negative charge, or anything ionized, anything that has full units of charge, like the sodium chloride we saw last time. anything like that is going to dissolve in water. Even something big, like a protein, typically the surface of proteins are going to have regions that are polar. So proteins will dissolve in water. Anything with that property will dissolve in water. We call that hydrophilic. You break that word down, hydro means water, and philic means loving. So anything that has an attraction for water will dissolve. And that's a lot of things, and a lot of things specifically in biochemistry, as we'll see. So what's the opposite of hydrophilic? Hydrophobic. Those things will not dissolve. There are limits to this. Water is an extremely versatile solvent, but it can't dissolve everything. If something is hydrophobic, it will not dissolve. So these are things that are neither polar nor ionized. So they have no partial or full charges, and that means they can't form hydrogen bonds. No H bonds means you can't dissolve. So these are going to be things like oils, for example. So you can take oil and water, shake it up like you make like a salad dressing. You can shake up that oil and vinegar salad dressing, and it'll mix temporarily, but if you let it sit on the table for even just a few minutes, they're going to separate out. And before you pour it on your salad, you have to shake it up again. Otherwise, you're just going to get the oil on the top. You won't get any of the rest of it. All right? It's because the oil is hydrophobic, so it doesn't form those H bonds, so it separates out from solution. It doesn't dissolve. And those are the emergent properties of water. We did hydrogen bonds, cohesive behavior, temperature moderation, expansion when it freezes, and versatility as a solvent. And those are the emergent properties of water, and these are going to play a major role in a lot of the processes that we talk about over the next 2 semesters. Because again, all biochemistry takes place in an aqueous solution. Everything happens dissolved in water. So whether something is hydrophobic or hydrophilic is extremely important for what it can do, what it can interact with in that context. That's why we're spending time on this. Everybody okay? Questions? Yeah, go for it.
Can you explain capillary action?
Capillary action is when you have a, I guess the right word would be if you have a matrix of a substance that is hydrophilic. So like, a good example of this is just a paper towel, right? The fibers are hydrophilic in a paper towel. If you put a part of that object in water, I get a lot of practice with this because I have 2 small kids that spill something, always cleaning it up. If you have like spilled juice, apple juice, to take a completely hypothetical example I haven't experienced 9 times in the last week. If you take a little bit of spilled apple juice and you put a paper towel partially in it, the apple juice, because it's mostly water, kind of gets like sucked through the whole paper towel. That's capillary action. And it's because the material of the towel, the fibers, is hydrophilic. So as the molecules move, they're going to tend to move further into that substance because it attracts them. Yeah, water traveling up the xylem of a tree, yes. The vascular tissue of trees is another example of that, absolutely. And we're going to talk about that in quite a bit of detail next semester. We will do a little bit of plant physiology. Yeah, good job. All right, so now we can get to today's topic, and we are to the minute exactly at the same place we were this morning, which is really fun. So let's talk about biological molecules. We only have 2 big topics today, 1 and 2. So when you're doing your biostorm, first level of the outline, it's going to be 1 and 2, and then we'll have, A, B, C, D, however many in each one. Today, kind of in terms of how it relates to what we've been doing, is we're going up a level in complexity. So last time, talking about chemistry, we talked about atomic structure, and then today, and then we went up and talked about molecules, and now today we're going a step further. We're going to talk about the biological macromolecules, the big molecules that participate in all different biological processes. So in part 1 today, we're going to talk about carbon, and we're going to focus on the 7 functional groups, so compare and contrast functional groups. And then in part 2, we're going to talk about the types of macromolecules, we're going to start with the two types of reactions that allow us to build them and break them down. So sequence those processes of polymerization. And then we're going to compare and contrast the four classes of biological macromolecules. Those are our big outcomes for today. We're going to start with carbon. Because remember, it's all about carbon when we're doing biology. That's organic chemistry. That's why we call it organic chemistry. It's because living things and living systems involve chemistry that's based on carbon compounds. So it's always going to be C bound to more carbon and also to hydrogen. That's your kind of starting point. CH, you need carbon and hydrogen to have your organic compounds. We study this stuff. This is the study of living systems, right? Cells are going to be 70 to 90% water, and most of the rest of what we find in cells is going to be carbon-based compounds or organic compounds. This is why all the life science majors have to take organic chemistry, even if you're like, oh, I'm never going to do chemistry. You have to take organic chemistry because none of the biochemistry makes sense outside of the context of organic chemistry. I promise you. Organic chemistry is relevant to what you are doing. I promise you it is. And this is why. Because we're going to see examples of different molecules and different reactions, and to really understand how those reactions happen, why those reactions happen, you need to have an understanding of organic chemistry. It's not thrown into the bio major or the life science majors as a readout course. That's not the purpose of organic. I can't believe, as somebody who had to go through organic chemistry, I'm standing up here defending organic chemistry's honor. But like, seriously, it's useful. You gotta learn organic chemistry. Like, it's good. Once you get it, it's awesome. It takes time, but this is why you gotta do it. It's really important. Biochemistry, biology, it's all about organic chemistry. Just something I'm sure everybody's just thrilled to hear. Okay? Now, carbon is the big winner here. The reason this is carbon-based chemistry instead of oxygen-based chemistry or nitrogen-based chemistry is that carbon can form 4 covalent bonds. And this allows carbon to build big, complex molecules. Right? So if you think about a carbon atom, right? You've got a chain of carbon, right? It's hanging on to one carbon atom on this side and one carbon atom on that side. Okay, well, I can still form 2 more bonds. You can attach 2 more things to that carbon atom. Whereas with oxygen, you got a carbon on this side, carbon on that side, I'm done. Those are my 2 bonds. I only make 2 covalent bonds. I can't build any bigger than that. Right? So that oxygen, either it's kind of a dead spot in the middle of the chain that you can't add anything to, or maybe it's like the carbon to the oxygen and when you add something interesting onto that, you know, like a phosphorus atom or something. But now it's a dead end. That's the end of the chain. Right? Because now there's nothing else you can add. You can't add a carbon to that oxygen, so you've got 2 things on it. Carbon, by making 4 bonds, allows it to be the backbone for very complex molecules. Now, the simplest kind of carbon-based compounds are called hydrocarbons, which, as you can imagine, contain hydrogen and carbon and nothing else. And the simplest hydrocarbon is pictured right here. It's methane. You don't have to memorize that. I will tell you when you need to know like a specific chemical formula or a specific chemical structure. This is not one that you need to know, but just as an example of a hydrocarbon, this is methane, C with 4 hydrogen atoms. Now, hydrocarbons are nonpolar and non-charged, which means they are hydrophobic. So hydrocarbons do not dissolve in water, they don't form H bonds, which means biochemically, they're kind of, I won't say they're inert, they still do important things, but it's very context specific. There's lots of other compounds that we'll see will react in a lot of different ways. Hydrocarbons tend to be a little more limited because they're hydrophobic. Now, what we can do to change their properties, we can add functional groups. Basically, you take one of the hydrogens, or more, and you replace them with something else. Replace it with an oxygen or nitrogen, some other stuff. Once you do that, now you've changed their properties. Like you start adding oxygens and nitrogens all over the place, it's not going to be hydrophobic for very long. It's going to be hydrophilic, and now those properties are going to be different. It's going to interact with different things. It's going to participate in different types of reactions. So our baseline is hydrocarbons, but we can use functional groups to change their properties. Go ahead.
What do you mean by functional groups?
What do I mean by functional groups? I'm glad you asked. Thank you. Functional groups are some other group of atoms that will replace a hydrogen attached to a carbon in a hydrocarbon. So it could be an oxygen and some other stuff, it could be a nitrogen and some other stuff, something other than just a hydrogen attached to that carbon, and it's going to change the properties. And that means it's going to change the function. It's going to change what it can interact with, what reactions it can do. Remember, in biology, structure dictates function. So you change the structure of the molecule, you're going to change the function of that molecule. I'll give you a simple example here. Again, I'm going to show you two molecules. You don't need to memorize the structures. It's just to illustrate the importance of functional groups. All right, so let's look at these two molecules right here. These are both steroid hormones that humans use. Okay, you've got on the left estradiol, which is a form of estrogen, and on the right you have testosterone. Now you can see these structures are very similar to each other. You've got these rings, like 1, 2, 3, 4, 1, 2, 3, 4. You've got a couple things up here that are the same. The only differences are these functional groups that are highlighted in this pale blue here. All right, you've got one functional group here, and then you've got there and there. Those are the only differences between these two molecules. But physiologically, estrogen and testosterone are very different from each other, right? They have very different physiological effects. Why is that? Because they have different functional groups. The backbone is the same, right? It's the same 4 rings. That's the same. That doesn't change. They're carbon and hydrogen backbone. But a few of the functional groups are different, and that changes the properties. And that's why we care about functional groups. You change the functional groups, you change the properties, you change the properties, you change the function within a biological system. That's why this matters. So we're going to do here, we're going to go through 7 functional groups. For each functional group, here's what we want to do. We want to get the chemical formula, right, just what atoms are involved. I want you to know the structure. You need to know the structure, because we can give you a question and show you a molecule and say, like, what functional groups are in this molecule? So you need to know the structure. You need to know the properties, the big one being, is it hydrophobic or hydrophilic? Right? Is it polar or non-polar? Acidic or basic? Right? You need to know the properties in terms of how it will interact with an aqueous solution. Then, not today, for all of them, but eventually for each of the functional groups, we're also going to see examples of what they do in biological systems. We're not going to do that today for all of them, but eventually we are going to see examples of all of these in use as we go through other topics. So today, again, as we go through these functional groups, the chemical formula, the structure, and the properties in terms of being hydrophobic and hydrophilic. Like acidic, basic, charged, non-charged, that stuff. Yeah?
Are hydrocarbons a functional group?
Hydrocarbons are not a functional group, but one of the functional groups is a hydrocarbon, if that makes sense. Don't worry, we'll get there. Don't worry. That was confusing, I know. Hydrocarbons are a type of molecule. One of the functional groups is a hydrocarbon, but only one. So let's talk about our 7 functional groups. Our first one is a hydroxyl group, which is OH. It's an oxygen atom and then a hydrogen atom. It's OH right here. Now the types of molecules that have hydroxyl groups, they are called alcohols. And the name ends with ol, O-L. It's typically how the name ends. So like methanol or ethanol, which is pictured right here. You don't need to know the structure at all. Any of the structures I show you, they're just examples. You don't need to memorize the structures. I just need you to know the formula and structure of the functional group itself, but not the rest of the molecule I show you, right? So there will be examples here. So like this is ethanol. That's the alcohol that's present in alcoholic beverages. Most of you are too young to know anything about that, but you'll learn in a few years. I'm not stupid, that was a joke. So again, it's oxygen bound to hydrogen. That means, think back to when we talked about bonds, that is a polar covalent bond. Right? So that means that this functional group is going to be hydrophilic, because you're going to have partial charges on those atoms. Okay, so relating this to what we talked about previously, this is going to be a strongly hydrophilic functional group because it is polar. Okay, it's strongly polar. Okay, so that's hydroxyl group. That is functional group number 1. Functional group number 2 is the carbonyl group. This is a carbon with a double covalent bond to an oxygen atom. So it's C double bond O is typically the way you say it. It's just CO is the chemical formula. CO. It can occur in the middle of the molecule. It can occur at the end of the molecule. We don't care about the difference. It doesn't matter. If it has a C double bond O on a carbon, that is a carbonyl group. By the way, you'll also hear people pronounce this carbonyl group. It's the same thing. The way you say it is the way that you heard it the first time. So I was taught it as carbonyl group. That's how I say it. My brain is never going to rewire that fact, but say it however you want. Nobody cares. So this functional group is polar and hydrophilic, but it is not exactly as strongly polar as the hydroxyl group. The reason being, the oxygen and the carbon are closer in electronegativity than an oxygen and a hydrogen. So it's still polar, you still get a partial negative charge on that oxygen, but it's not as strongly so. It's still enough that it's going to be hydrophilic, it's going to dissolve, but there are shades of gray when it comes to the degree of polarity that we see in these functional groups.
Is there supposed to be a C up there?
See, yeah, I mentioned double bond O. Either, yeah, C double bond O. As long as you double bond O is the critical part. Yeah. Yep.
Can you explain again why it's not super?
It's not as polar because the difference in electronegativity between C and O is smaller than the difference between H and O. So that oxygen is still pulling just as hard, but carbon resists more. So the difference is smaller. All right, that's our second one. Now our third one is kind of a combination of the first two. You take a carbonyl group and a hydroxyl group and you put them on the same carbon atom and you get a carboxyl group, which is COOH, but that's not the structure, that's just the formula. It's carbon atom with a double bond to an O and then a single bond to an OH. Okay, so it's COOH. Now this is really cool because you put those two things together, the OH and the double bond O, and that leads to emergent properties because this group has a property that is absent from the other two groups. And that property is that it's acidic. What do we mean by acidic? It means this hydrogen right there, and specifically the proton, that is the nucleus of that hydrogen, that will dissociate in an aqueous solution. But the oxygen, because it's so electronegative, will keep the electron, so the oxygen that's left behind becomes negative. So you end up with this, where in biological systems, you actually end up with the COO minus, is what you tend to see. That's the ionized form. And it releases that proton into solution, and that's acidic. That's what being an acid is. So in its neutral form, over here, right, it's polar, and then its ionized form, it's ionized, it has a full unit of charge. Either way, obviously, this is going to be very strongly hydrophilic. We're going to see this when we talk about proteins, because proteins are made of things called amino acids. The acid part of the name in amino acid is because every one of those building blocks has a carboxyl group on it. So let's pause here for a second. What questions do we have about any of the first three? Hydroxyl, carbonyl, or carboxyl? Any questions about those three? Yeah, right. How do you know if it's hydrophilic or hydrophobic? If it's polar or acidic or basic, right? Either if it's partially charged, it's polar, or if it ionizes with a full charge like this, that means it's hydrophilic. If it's neither of those things, it's hydrophobic. And all of the ones we're going to look at today are hydrophilic, except the last one. Yeah, so this OH bond right there, again, it's an emergent property due to the influence of what's going on over here. We're not going to get into the chemistry of it, but basically that, having the two oxygens pulls the electrons away from the hydrogen. So it's a very weak bond. So in an aqueous solution, forming H bonds with water, that affinity is greater than the affinity that that proton has for the oxygen, so it tends to dissociate. You don't need to know that level of detail, don't worry. All right, functional group number 4 is the amino group, which is NH2. Right, so nitrogen with 2 hydrogens bound to it. Now this, in terms of its properties, is the opposite of a carboxyl group, because this one is basic. That means that it will, rather than lose a proton, it'll actually gain a proton from the environment. So in aqueous solutions, in biological conditions, what we tend to see with amino groups is they're going to be in their ionized form of NH3+. Right? So instead of donating a proton into solution, they accept a proton from the surrounding solution and become NH3+. Obviously, if you have a charge, you are hydrophilic. So these are going to be hydrophilic. Even without the charge in a neutral form, they're still going to be polar because the NH bonds are asymmetrical in terms of their electronegativity. So those are polar covalent bonds. So in either form, they're going to be hydrophilic. We tend to see them in an ionized form under biological conditions. This is the other component of amino acids. So that name for that molecule, amino acid, it's acidic because of the carboxyl group we just saw, and it's basic, and the amino part of its name comes from the presence of the amino group. So that's number 4. 3 more. The next one is actually pretty simple. It's a sulfhydryl group. This is the same as the, question, go ahead. Everything so far is hydrophilic. If it's polar or acidic or basic, it's going to be hydrophilic. Sticking with that theme, the sulfhydryl group is the same as the hydroxyl group, except it's sulfur instead of oxygen. Sulfur is heavier than oxygen, but it has the same electron configuration, so it forms 2 bonds just the same way. This is a component of some amino acids, and even though it's sulfur instead of oxygen, it is still going to be weakly polar. We're still going to call it polar. It's weakly polar. It's just barely polar. But for us, it's still polar. We're only going to see this in a few very specific circumstances, like there's a type of bond that forms in proteins that we'll see when we do proteins, called a disulfide bridge or a disulfide bond. That involves sulfhydryl groups. So we'll get there when appropriate. For today, I just want you to know that the structure is SH, so it's sulfur instead of oxygen, like hydroxyl group, and then it is weakly polar. It's the least polar of the hydrophilic things that we're talking about. It's very small, only has 2 atoms. Question?
Sorry, this is about the amino group, but how do you know that that's basic?
How do you know that it's basic?
Yeah.
Just you would biochemically detect that it just absorbs the protons and exists in its charged form. That means it has to be basic. It has to get that positive charge from somewhere.
Can you repeat how you find out if it's hydrophilic or hydrophobic again?
Anything that is polar or charged is hydrophilic. And if it is neither of those things, then it's hydrophobic. Another way is if it has atoms other than carbon and hydrogen, it's hydrophilic. All right, number 6 is the big one, phosphate group. So its neutral form, the chemical formula for this is PO4H2. So it's a phosphorus atom, 4 oxygens, and 2 hydrogens. But you never see it that way under biological conditions, because a phosphate group is even more acidic than a carboxyl group. It's very strongly acidic. So those 2 H's, they dissociate. Those protons leave. What you always see is this, where it's P with 4 oxygens, and 2 of them have negative charges. Now, the way that's typically written is kind of weird. It's like this. It's OPO3 2 minus. The reason for that is it's connected to the carbon backbone via an oxygen. So if you start with the carbon, it's OP3 O3 with your 2 units of negative charge on there. And again, the reason for that is these two oxygens in neutral form would actually be OH. But those are very weak associations, so those protons just pop off, the oxygens keep the electrons, so you get 1 unit of negative charge on each. Okay, now we're going to see phosphate groups all over the place. We're going to see phosphate groups in DNA and RNA, we're going to see phosphate groups in ATP. These are everywhere, so we're going to get to the specific context for them down the road. We're going to see a lot of phosphate groups over the next, not just the next couple of weeks, but over the next 2 semesters. Don't worry about like where we're going to find them right now. We're just going to hit those different molecules as we get to them. Okay, we have one more, then I'll pause and we'll take questions on the back 4 here. So we have one more after the phosphate group, and this one is very unique. We haven't seen anything like this last one before, because so far, none of our functional groups have been hydrocarbons, which means every one of them has been either polar, or basic, or acidic. Which means so far, all 6 have been hydrophilic. Our last one is a methyl group. The formula for methyl group is just CH3. It's a carbon with 3 hydrogens. Now that, as you can tell, is just a hydrocarbon. Which means those are nonpolar bonds, those are nonpolar covalent bonds, which means, unlike the other 6 we just talked about, a methyl group is hydrophobic. It does not dissolve in water. Things that are methyl groups are not going to dissolve in water. It has to have something else in order to do that. Okay, so methyl groups are unique because they are hydrophobic. Now, we're not going to see too much of methyl groups over the next couple of semesters. They're not like phosphate groups or anything. Phosphate groups, hydroxyl groups, carboxyl groups, those things are everywhere. Methyl groups are a little more limited. We're really only going to see them in one place. It's when we talk about regulating gene expression. Somewhere in October we're going to do that. Who has heard the term epigenetics? Anyone heard that term? Cool. So a lot of what is going on with epigenetics is you're adding methyl groups to DNA, methylating the DNA. You don't need to write that down. We're going to talk about it in next month. Don't worry about it right now. But like just as a preview, that's where methyl groups are going to come into play. Because methylating your DNA is a way to regulate the level of gene expression in that particular region of your genome. So we're going to get to that later in the semester. That's just kind of so you have an idea of where we're going to see these, because we're not going to see these for a little while, unlike some of the other ones. So what questions do we have about the back 4? Amino, sulfhydryl, phosphate, or methyl? Any questions there? If it's hydrophobic, it's automatic. If it's hydrophilic, it either has to be polar or acidic or basic. Like it has to either get a partial charge or a full charge in order to be hydrophilic. And if it's nonpolar, it's automatically hydrophobic. Those words always go together. Okay, no, it was a lot. Oh, yeah.
When you say acidic and basic, you mean partial polar and partial non.
When we say acidic or basic, we mean that it'll either the protons will dissociate from it and it will keep a negative charge, so that's acidic, or it will absorb protons and acquire a positive charge. That's basic. And once it does that, it ionizes, it has a full unit of charge, and that of course makes it extremely hydrophilic.
That's actually a shortcut. If you were not sure which one's acid or basic.
You can tell if it's
Acid or
charged
basic.
because if it's either acidic or
Basic,
basic,
whereas
it's either
basic
going
is
to lose
not.
protons or gain protons. And that's what causes it to be charged. So if it's
Oh, no.
either acidic or basic, you're going to be charged, and that makes you hydrophilic. If it's just polar but not acidic or basic, then it won't require a full charge. It will have a partial charge.
So like in this image, it said that it's bonded to like 2H. So then like
You'll be there and there.
yeah, so like what is the negative sign exactly represent that?
The negative sign means that the proton, so a hydrogen atom is 1 proton, 1 electron. What happened there is the proton left, but you kept the electron. So now you have 1 more unit of negative charge compared to the positive charges, so you have a net negative charge. And each oxygen has 1. All right, last one, we're going to keep going.
I just have a question about the non
So polar is where the bond is asymmetrical, so you get partial charges. That makes you hydrophilic. If you're nonpolar, like these bonds right here, that means those bonds are close enough to symmetrical that neither side of it gets a partial charge. That means it's hydrophobic,
Which is not cool.
which is nonpolar. Yeah. Okay, so functional groups. They're chemical groups that will replace the H in a hydrocarbon. That affects the chemical properties of the molecule, and if it affects the properties, it affects the function. That's the big idea with functional groups. All 7 of those that we saw, obviously there are more than 7. Those are the 7 we care about. We're going to see these in all different contexts throughout the rest of the semester. Some of them we're going to see today, some of them we're not going to see for a little while. But we're going to see all of them as we go through the next 2 semesters. So now let's talk about biological macromolecules. My watch battery is dead, so I don't know what time it is. How are we doing? All right, yes. Where's me? So biological macromolecules. This word macromolecule is just a large molecule. In other words, like thousands of atoms, sometimes bigger, tens of thousands, hundreds of thousands of atoms in a single molecule. Most macromolecules that we're going to deal with are also polymers. A polymer is a chain of repeated small subunits called monomers. Monomers are just the identical or very similar building blocks for a polymer. So poly means many, mono means one. So this is the singular pieces, this is the chain of those repeating pieces. So like some polymers, for example, DNA is a polymer. For each nucleotide, each base, that's your monomer. So it goes long chain, right? Not everything we're going to see today is a polymer, but most of the macromolecules are polymers. There are 2 chemical reactions that we're going to start with before we get to the 4 types of biological macromolecules. There are 2 types of chemical reactions. And it's really important that we do the reactions. Because if we don't do the reactions, a lot of what we do down the road, whether it's DNA synthesis or photosynthesis or respiration, none of that stuff's going to make sense. So we're going to do the reactions first, then we're going to get to the 4 classes of molecules. Okay, so our first chemical reaction is what we use to build macromolecules, and that is the dehydration reaction, also called dehydration synthesis. And it's called dehydration reaction because the net effect is to build up your polymer by removing a molecule of water. We can see that happening right here. You have a short polymer with 3 monomers in it. You're going to add a new one. You've got a hydrogen and a hydroxyl group right here. They're going to react together because look at that, H2O. They're going to react together. You're going to remove some water. And you're going to form a new covalent bond between the existing polymer and the new monomer. Right there, you're going to form that new bond. Okay, so that's called the dehydration reaction, and we use this reaction to build up bigger molecules.
Would that be like condensation?
I think condensation reaction is another word for this. Yeah, I always know the term dehydration, but condensation also because you get water out of it, basically. Same thing. The enzymes that accomplish this are called dehydrogenases, which is nice, because dehydration reaction, dehydrogenases. Anytime you see the suffix ACE, A-S-E, ACE, that means the thing is an enzyme. Now, we haven't defined the term enzyme yet. We're going to do that more technically in a couple weeks. For today, an enzyme is a protein that speeds up a reaction. That's all we need to know. So the enzymes that do the dehydration reaction are the dehydrogenases. Now, will anyone know what the opposite of a dehydration reaction is? What's the opposite? Just shout it out if you know it. Yeah, it's hydrolysis. Good. It's the exact same reaction, just going in the opposite direction. It's exactly the same thing, just reverse. So in this one, we're going to take our polymer, we're going to split a molecule of water, we're going to add a hydrogen to one monomer, we're going to add an OH to another monomer. And in doing so, we're going to break that covalent bond between those 2 monomers. So a hydrolysis reaction results in taking a bigger molecule and making it smaller instead of the other way around. Smaller molecules and make them bigger. We call this hydrolysis because of this step right here. Let's break this word down. Hydro means water. Lysis means splitting or breaking. So we have water splitting is how we accomplish breaking this bond. We split a molecule of water. So the type of reaction is called hydrolysis. Anyone want to guess what the type of enzyme is that promotes this reaction? Hydrolases. Just add ACE. Hydrolases do hydrolysis reactions. And we're going to see lots of examples over the next few weeks, especially once we get into photosynthesis and respiration. We're going to see examples of both hydrolases and dehydrogenases. We're going to see both of those as we go through the different energetic processes. And so that's hydrolysis and dehydration. Do you have any questions about those? Okay, so that's how we get our molecules. Let's talk about our macromolecules. The four classes of macromolecules are carbohydrates, lipids, proteins, and nucleic acids. Lipids are unique because they are not polymers. In other words, lipids are not repeating chains of monomers. The other three are, as we'll see. Okay, so carbs, proteins, and nucleic acids, those are all polymers. In this lecture topic, one, we're not going to get through all this today, and that's fine. We did it this morning. That happens every year. Like I said, we're continuing the tradition, and that's cool. So we're probably just going to do carbohydrates and lipids. Of course, now that I said it, I changed myself, but whatever. And then nucleic acids, we're basically not going to do it all. It's one slide. All it says is like DNA, RNA, we'll do it later. So like, don't even worry about this right now. We're going to do this next over. So let's start with carbohydrates. We'll talk about their properties. Then we'll get into the rest. We'll see how far we get. Yeah, I don't have a clock. You don't realize how often you check your watch during class, and so you don't have to. So you need to change your watch battery. All right. So let's start with carbohydrates. Carbohydrates are made of three elements: carbon, hydrogen, and oxygen. The ratio is usually somewhere in the neighborhood of 1 to 2 to 1, CH2O, 1 to 2 to 1 ratio of carbon, hydrogen to oxygen. Carbohydrates are big polymers, big long chains. The monomers are sugars, otherwise called monosaccharides. Right, so the sugars, the monomers, are going to have 3 to 7 carbons each, and every one of those carbons is going to have a functional group on it. Most of them are going to have hydroxyl groups. One of them is going to have a carbonyl group. And because those functional groups are all polar, that means that sugars are extremely hydrophilic. Sugars very readily dissolve in aqueous solutions. This is why, if for some reason you ruin your coffee with sugar, when you stir it dissolves. It doesn't just sit in a big clump at the bottom of your cup of coffee. I don't know why anyone would do that, but if you do that, I'm not judging you, just saying you're ruining your coffee by putting sugar in it. If you do that, it dissolves. And it's because sugars have all these hydrophilic functional groups, so it very readily forms H bonds with water. Those sugar crystals break down. And so carbohydrates are made of sugars, and it's all C, H, and O. You okay? All right. Functions of carbohydrates, they are for energy and structure. And we're going to see examples of that in just a minute. When we talk about specific examples, we'll see some that do each. So you either break down your carbohydrates for fuel, or some carbohydrates are actually really hard to break down, and they form things like cell walls in plants, and also in fungi. And just in terms of a naming convention, we just talked a minute ago about how enzymes end with the ACE suffix. Sugars tend to end with the OS suffix. So like glucose, sucrose, fructose, lactose, those are all sugars. That tells you what they are. If you see OS, you're dealing with the sugar. It's either going to be a monomer or it might be a dimer, and we'll talk about the names for those in a minute. Okay, so just anytime you see OS, you don't have to write that down. I'm not going to ask you about that. But anytime you see OS, you should know that thing's a sugar. Just like anytime you see ACE, that's an enzyme. So the monomers those sugars, the technical word for the sugars are monosaccharides, one sugar. And this table, don't like draw these. This is just to give you an example of like what the different structures look like. You can have different numbers of carbons, 3 or 5 or 6. You have different locations of the functional groups, right? It could be at the end, it could be in the middle, doesn't matter. We classify them based on the length and the location of the carbonyl group. This is all just FYI for you. You don't have to know any specific sugars, except glucose is going to be important. Other than that, you don't need to know any of these structures. I'll show you glucose in a minute. That one's going to matter. The rest, like, you don't need to know structures. And for glucose, it's the formula, not the structure that matters. When you get to like biochem, you're going to need to know some of these in more detail. Like, you're going to become really good friends with like ribose when you take biochemistry. But for today, just want to show you some of the diversity of the structures. That's all we're doing here. Okay, so monomers of your carbs are monosaccharides. Now, glucose is the most common monosaccharide. And I do want you to know the chemical formula for glucose. That's going to be C6H12O6. You need to know that chemical formula. Now, the rest of this slide is not something I'm going to ask you about, but it is useful information to have in terms of why certain carbohydrates have certain functions. Okay, glucose is pretty diverse in terms of the ways we find it. You can have a linear form of glucose like this, where it's just the carbons 1, 2, 3, 4, 5, 6 all in a line. You can also have a ring form of glucose. It'll close up and form this ring. And there's two different ways that can happen. The word that I want you to know that's relevant here, it's going to come up a few more times over the course of this class. The word is isomer. You can form two different isomers of that ring version of glucose. An isomer is when you have one molecule, but it has two different physical conformations. Same atoms, same structures, all the same atoms are connected to each other, but the orientation can be different for some of those bonds and such. And those are called isomers. Now, you don't need to know the specifics of like alpha and beta glucose and which one's different, why they're different. You can see that the two hydroxyl groups for alpha are on the same side of the ring. For beta glucose, they're on opposite sides of the ring. You don't need to know that. Like, this is just showing you the variety here, because when you make chains of the different types of glucose, that's going to affect what you can do with those chains. That's all that matters right here. Is everybody okay with that?
Are there isomers important to orientation?
Are there isomers of more than two? I'm sure there are. I can't think of an example because biochemistry was a long time ago. But I'm sure there are examples where something has more than two isomers. Yeah, I'm sure. That I would be shocked if that wasn't a thing. Why are the carbons not labeled on the ring structure? Oh, down here? I don't know.
So even isomers is something like a molecule that has different ways to
Yeah, so same molecule, different physical orientation. And that can affect the properties. But again, you don't need to know the specifics of this. I just want you to know the term isomer. As long as you have that concept, you're good. Okay, so let's go up in scale to disaccharides. Let's go one step up. Disaccharides are when you have two monosaccharides linked together. Now this is important because there is a characteristic type of bond for each of the macromolecules we're going to talk about. The characteristic type of bond for disaccharides is called, or for carbohydrates, is called a glycosidic linkage. I'm going to show you an example of reaction forming a glycosidic linkage. You don't need to know this specific example. What I want you to see is, one, the type of reaction that it is. You probably guess what it's going to do. And two, the resulting molecules so that you can recognize, oh, that's what a glycosidic linkage looks like. Okay, so here's the reaction. This is making sucrose, which is table sugar. This is taking glucose and fructose and linking them together. The key thing here is this is an example of the dehydration reaction that we just talked about. So you've got your OH right there, you got your H right there, you're going to pull out that water molecule, and as a result, the remaining oxygen, that O, right here from this side, that's going to form a new covalent bond with this carbon all the way over there. So now you have your one monomer right here, you have your other monomer right here, you have a glycosidic linkage where it's just this oxygen in the middle where the oxygen is hanging on to the two rings on either side of it. That's a glycosidic linkage. You need to know that word, glycosidic linkage. When you're making a table, doing your compare and contrast biological macromolecules, that term, right, you can have one of your categories, type of bond. And glycosidic linkage is going to be the type of bond for your carbohydrates that links your monomers. Okay, so that's what we care about right there. Yeah?
It's always held together by water as a base.
It's not that the water holds it together, it's you remove the water, you do dehydration synthesis, and what's left behind makes the bond. So it's the oxygen in this case. And each thing that we look at, each type of macromolecule is going to have a different type of bond that's characteristic of that type of molecule. For carbohydrates, it's a glycosidic linkage. All right, so you go up in scale. Mono is 1, di is 2, poly is many. So polysaccharides are chains of hundreds, if not thousands, of monomers, monosaccharides. Now, polysaccharides generally do one of two things within cells. They can either be for energy storage, usually like short-term energy storage. So this is going to be like starch in plants or glycogen in humans. You probably don't know what glycogen is, that's okay. We're going to talk about it a lot next semester. It's stored in your muscles and your liver, and it's related to your blood sugar. So it's how we store sugar in our body. And plants do it with starch. But you can also have structural polysaccharides, which is kind of weird because we don't think of sugars as particularly like structurally robust molecules, but they are as long as the components and the bonds are the right kind. So cellulose, like starch and glycogen, is just a chain of glucose. Now I always forget which is alpha, which is beta. If anybody knows, feel free to tell me. I always forget which is which. But the bond angles between the monomers and cellulose is such that it's really hard for enzymes to get in there and break them apart. Whereas it's really easy to break down the monomers, right, from either starch or glycogen. Happens all the time. You just use that energy, that's the point. Cellulose is going to be like one of the materials that makes up the plant cell wall that we'll talk about in a few weeks when we talk about cell structure. That is very robust, and there aren't many organisms on Earth that make the enzymes to break that down. It's just a polymer of glucose, just like the other two, but it's the opposite isomer, so the bond angles are different between the monomers, and the enzymes can't break it down. And then chitin is a really interesting one. Chitin is actually a material that makes up like crustacean shells, and also the cell walls and fungi. And you probably haven't heard of chitin, unless like you've ever played StarCraft, in which case you've probably heard of chitin because there's one science fiction real-time strategy game with aliens, and they get chitin shells. I'm such a nerd, don't worry about it. It's a great game. Anyway, the function is determined by what it's made of, and then the position of the linkage, as I was saying, with cellulose compared to the other two. It's just a different bond angle, and the enzymes either can or can't break it down. Again, structure dictates function. One of the big themes that we have going on here. How's everybody doing? Feel okay with carbohydrates? Chains of sugar, different functions based on the structure. Doing great. You should be up to get carbohydrate summary. So carbohydrates are carbon, hydrogen, and oxygen. The monomers are sugars, also called monosaccharides. They're going to be energy storage, usually short-term energy storage, and they can also be structural, depending on the chemical structure. Here's our carbohydrates. So the last thing we're going to talk about today is the lipids. We'll get through that. Yeah. What's the purpose of the glycosidic linkage? It's not so much that it has a purpose, it's just the type of bonds that forms when you're linking monosaccharides together. That type of chemical bond is called a glycosidic linkage. And when we do other things, we're going to see other ones. So like in the lipids in a minute, we're going to see an ester linkage. It's just a different type of chemical bond that links the two things together. When we do proteins, it's peptide bonds. So it's just each type of molecule has, hey, who's ever talking over there? I can hear you all the way over here, which means so can everybody else, so that's distracting, so we'll stop. Yeah. Always covalent bonds. Everything we're talking about in here, yeah, covalent, absolutely. Okay, so let's talk about lipids. Now, lipids are a really diverse group of molecules. We are not going to talk about all the different types of lipids that are out there. We're just going to cover 3 big ones today. They share some traits in common that are really important. Because lipids are mostly made of hydrocarbons, they're going to be hydrophobic, which is going to be different from all the other types of macromolecules we talk about. And they're also different in that they are not polymers, meaning the other 3 types, the nucleic acids, the carbs, and the proteins, are long chains of repeating monomers. That's not the case for lipids. Lipids, you can make very big molecules, but they are ultimately all dead ends. So you can't just make an infinitely long chain like you can with the other stuff. So because they're hydrophobic, they do not dissolve in aqueous solutions, like the inside of a cell, for example, or your bloodstream. So this obviously is really important in terms of the role they play in biochemistry. But they can dissolve in non-polar solvents, something like chloroform, for example. So if you've ever done like a DNA extraction from a cell in a lab, one way to do that is called a phenylchloroform extraction. You use a non-polar solvent, chloroform, to dissolve the lipid membranes, and that allows you access to the DNA. Right, so the properties are quite different from some of the other things that we've talked about. Do you need to know that that lipids will dissolve in chloroform? No. Do you need to know that they are hydrophobic and will not dissolve in water? Yes. That is the key thing. Like, that is the big thing when it comes to lipids. So we're going to talk about 3 lipid families. We're going to cover fats, we're going to talk about phospholipids, and we're going to talk about steroids. Most of what we're going to talk about is fats, and then the phospholipids and steroids, they get 1 slide each, and they're each going to be important later on for various reasons. So we're going to spend more time on each of those down the road in various topics. But for today, there's not too much to talk about there. So let's start with fats. Fats are the most abundant lipid, and they're involved in energy storage, typically long-term energy storage. The reason for this is that they're way more energy dense than either carbs or proteins. Fats are approximately 9 calories per gram in terms of their energy. Carbs and proteins are only 4 calories per gram. So in terms of like bang for your buck, you get a lot more energy for a given volume if you're storing fats compared to something else. So these are long-term energy storage. Okay? Now the structure has 2 parts. The first part in gray right here, also don't like draw this molecule, don't do that. I'm just gonna point out the parts. You should be able to recognize eventually that this is a fat, but like, don't draw this, that's a waste of your time, don't draw it. So this right here, that part in gray, that is glycerol, which is a 3-carbon alcohol. In the absence of anything bound to it, it's just 1, 2, 3 carbons, and each of those carbons is going to have a hydroxyl group attached to it. Okay, so it's a 3-carbon alcohol with 3 hydroxyl groups. Those hydroxyl groups are where you can react, you can bind there. So the other components shown in gold are the fatty acids. These are long, unbranched chains of hydrocarbons. So it's just carbon and hydrogen, like 14 to 22 carbons. And then at one end, at this end, the acid end, you're going to have a carboxyl group, like we talked about before. And that's why they're called fatty acids. You have this long hydrocarbon tail, that's the fatty part, and then you've got your carboxyl group at one end, that's the acid part, so fatty acid. And a fat is when you take your glycerol and you attach 3 fatty acids to it. Now this is important because again, we want to compare and contrast all these different molecules. The type of bond that we use to connect these is called an ester linkage. Right in there. So each, in that red dashed box for each one, that is an ester linkage. So like in your table, type of bond, you've got glycosidic linkage for your carbs, You're going to have ester linkage for your fats or for your lipids. Yeah. Glycerol is strongly hydrophilic because of those hydroxyl groups, yes. Once you add the fatty acids to it, the long tails are kind of in the driver's seat. So it's going to behave hydrophobic despite the glycerol. That's only 3 carbons. Each one of these is going to be a minimum of like 14 or 15. So in terms of the amount of like polar versus non-polar regions going on here, overwhelmingly non-polar once you add the fatty acids to it. Good thing in the yes, the glycerol is going to be strongly hydrophilic prior to adding anything to it, yes. So let's zoom in on that because I should have asked you what type of reaction it is. What type of reaction is it? It's a dehydration reaction again. It's the same kind of reaction, just the difference is what the participants are. So you get a different type of bond as a result. Instead of a glycosidic linkage, it's this carboxyl group reacting with this hydroxyl group. You take out your H2O, and what remains is an ester linkage. So again, you should know ester linkage is what we find in lipids typically. Okay, ester linkage, lipids. It's still doing a dehydration reaction because again, you're going from smaller subunits to a larger macromolecule. In GenBio, basically any time you do that, you're going to do a dehydration reaction. Outside of GenBio, it gets more complicated. But inside of GenBio, anytime you go from small molecules to bigger, you should think dehydration reaction. That's what's going on here. Does everyone see that? Does everyone see another example of the same reaction? Okay. So that's how we build our fats right there. Now there are 2 main kinds of fats, and it's based on the type of fatty acid. There are saturated fatty acids, which is all single bonds between the carbons, single covalent bonds, which means they are full, they're saturated with hydrogen, because each carbon in the chain can fit 2 hydrogens on it. It's saturated. Now, saturated fatty acids, because there are these long, straight chains of hydrocarbon, they tend to be solid at room temperature. The reason for that is van der Waals interactions, specifically London dispersion forces that you probably talked about in chemistry. That's where, as electrons are moving, you have very short-lived regions of positive and negative charge, and if opposite charges appear next to each other, they're going to be attracted to each other. Very briefly, we're talking milliseconds. But if you have that happening up and down these chains all the time, when you have saturated fats together, they tend to stick together in a solid form. So like butter, it's largely saturated fat, solid at room temperature. We find this in like animal fats, except for fish. Fish tend to be the other. Right, but this is mostly going to be animal fats. And again, it's because they're these long straight chains, so you can pack them in very densely, and they're strongly attracted to each other due to the number of van der Waals interactions taking place. What's the other one I'm going to show you?
Saturated.
Unsaturated fatty acids. And basically the structure is a little bit different and means all the stuff is the opposite. So unsaturated fatty acids have double bonds. If you have a carbon that has a double bond, that means it has one less hydrogen. So like with single bonds, it's okay, I can hold on to this carbon, I can hold on to that carbon, and I have space for 2 hydrogens. If I'm holding on to this carbon and that carbon, I'm going to make a double bond. Now I'm 1 limb short. Now I can only have 1 hydrogen instead of 2. The other thing the double bond does is it puts a bend in the molecule. It kicks it out at an angle. So when you do that, you can't pack them in as densely. That means you have just fewer, you have less surface area for interacting. You have fewer van der Waals interactions. So they don't stick together as much. The specifics of these interactions are going to depend on the type of fatty acid. It could be monounsaturated, which is what we see here with 1 double bond, or it could be polyunsaturated. That means 2 or more. In this class, we're just going to call them all unsaturated. They have basically the same properties as far as we care. And the properties are that they don't associate with each other strongly, which means they tend to be liquid at room temperature. And the reason for this is if you have a bunch of molecules like this, you can't pack them in as densely. And if you can't pack them in as close to each other, because they're all kicked out at weird angles, then they're not going to have as many attractions to the London dispersion forces. So instead of being really stable, being solid at room temperature, they tend to be liquids. This tends to be like plant oils and fish oils. So what we tend to, where we tend to find unsaturated. One way you can think about the difference here is think about a room full of people where everyone's standing like this, everyone's like packed in really tight, versus a room where everyone's got their arms stretched out. Right? You can't get people as close together if your arms are all stretched out. That's what the unsaturated fatty acids are doing. So that's why you can't pack them in this gasoline. Now, I'll tell you, because somebody always asks, well, what's going on with trans fat? It's the same thing as unsaturated, but the bond angle, the double bond, is different. So they're in a straight line, and they're smaller because there's less hydrogen. You don't need to know, I'm just telling you because people always ask. It's the same thing, but instead of kicking out at an angle, it goes straight. But it keeps the double bond, which means fewer hydrogens, which means smaller diameter, so you can pack them in even more densely than the saturated fats. So that's what's going on with trans fat. In case you were curious, it's saturated both with a double bond. In terms of shape, it's saturated, but it has a double bond. The next type, so that was all fats. That was all fats. Our second class of lipid is the phospholipid. And there's only one slide on this because we're going to spend so much time on phospholipids down the road when we do membranes. For today, just know that it's the same thing as fats with one difference. It's a glycerol with 2 fatty acid tails, and then instead of a third fatty acid tail, it has a phosphate group. Now this is really important because that phosphate group we just learned is acidic. It's strongly hydrophilic, which makes that phosphate group different in terms of its properties compared to the tails. The tails are hydrophobic. So the word for that, where the molecule has regions with different properties, we call that amphipathic. It's got that hydrophobic head region with phosphate group, and it has the hydrophilic head region with phosphate group, and the hydrophobic tail region. Now, I don't want you to worry too much about details with phospholipids. Just know that it's glycerol, phosphate group, 2 fatty acid chains, and then it's amphipathic. Don't worry about beyond that, because it forms membranes, and we're going to talk about membranes in a lot of detail down the road. So we're going to revisit phospholipids and how they work and why they matter. But I just want to introduce the molecule today. Okay, so that's our second class. Our third one, the last thing we'll do today, is steroids. The only thing I want you to see here is the basic structure, and know that they can be involved in signaling, like hormones, or structure as part of the membrane. Okay, so steroids always have 4 rings. They have 3 rings that are 6 carbons, right here. They have 1 ring that's 5 carbons. And if you see this 4-ring structure, you know that's a steroid. I want you to just, that backbone structure, always steroid. That's what that is. So we talked about earlier, I showed you examples of the sex hormones, estrogen and testosterone. There's lots of other hormones that are steroid hormones. They're all built from this molecule, which is cholesterol. Don't memorize the functional groups, it doesn't matter. It's built from cholesterol, which is involved in membrane function, membrane structure. And we're going to talk about that when we talk about membranes. That'll be about a week and a half, 2 weeks from now. And that's all we need to worry about for steroids for now. Okay, so that's where we're going to end it for today. We'll start with proteins next time, and then we'll talk about the topic for next time, which is super fun. Now, here is your practice question. Remember, always highlight the outcome here. Compare and contrast macromolecules. Compare and contrast macromolecules. Which of the following macromolecules involves monomers that are sugars? So, take a minute, talk to each other, answer that.
I thought it was A.
And then if you have any questions, feel free to line up over here. What's the answer? A, starch, good. All right, thanks everybody. I'll see you on
Everything,
Thursday.
yeah,
Come on over here
protein,
if you have any questions. Thank you.
phospholipids are fat, C and D are fat, starch, glucose, there are carbs, no, it's A.
So, what does that mean? What is protein?
Protein is a bunch of amino acids. Yeah.